Clinical Chemistry Introduction to Amino Acids and Proteins. Amino acids are the building blocks of proteins. There are essential amino acids and non-essential amino acids. And in reference to human health, essential amino acids are those amino acids that are required in the diet. So these amino acids we actually have to consume in the diet because our bodies are not able to produce them or take other substances in our body and convert them to these amino acids. So these have to be supplied in our diet. Non-essential amino acids include those amino acids that we can produce ourselves. The general structure of an amino acid consists of this amine group, carboxyl group, a hydrogen, and then there is an R group that's attached. The R group is unique for each amino acid that exists. Recently, 2 new amino acids were identified, and they're listed here. Aminoacidopathies. So aminoacidopathies are talking about diseases that are related to amino acids. They're inherited enzyme defects that inhibit the metabolism of certain amino acids. And they can do this either by causing a dysfunction of the enzyme that actually metabolizes that amino acid, or it can do it by the membrane transport system so that the cell would no longer be able to transport that amino acid. Some states require this screening for up to 26 amino acids, and these are screened for at birth. Over 100 diseases are the result from aminoacidopathies. They can cause severe medical complications because of the buildup of toxic amino acids and the byproducts of amino acid metabolism in the blood. This is a list of some of the common aminoacidopathies. The 2 in bold are ones that you should make special note of, so phenylketonuria and maple syrup urine disease. Phenylketonuria we're going to talk about a little bit more on the next slide, but the maple syrup urine disease, just be familiar with the amino acids that are impacted here. So valine, leucine, and isoleucine are affected. And this is a disease of neonates, so it's usually diagnosed at birth. And they have maple syrup smelling earwax and urine. So lots of complications can be associated with this, but this is all you really need to know about maple syrup urine disease. It's just those amino acids that are associated. Phenylketonuria is an enzyme deficiency that causes increased phenylalanine in the blood and phenyl compounds in the urine. So phenylalanine is a pretty large amino acid, and this amino acid cannot be metabolized properly, so you start to see it accumulating in the body. For people with phenylketonuria, it's dangerous for them to eat protein or consume aspartame, which is an artificial sweetener. And this can cause neurological disturbances and brain damage. There is no treatment, and their only option is to limit eating phenylalanine in the diet. So they can't just consume large quantities of protein. They have to really consider everything that they're eating, and they can't regularly eat normal proteins that other people would be able to eat. There are specialized protein formulas that don't have phenylalanine in them, so those have been produced. Of course, they are expensive, but it's a way for them to get the protein that they need from their diet without having excess phenylalanine consumed. This is diagnosed using thin layer chromatography with ninhydrin. There's also a urine color test and the Guthrie bacterial inhibition assay. Basically with this assay, we're going to plate some Bacillus subtilis, some B sub, onto a plate of specialized medium. Then we're going to take a drop of the patient's blood on a filter paper disc, let it dry, put that filter paper disc on top of the surface of the bacteria, and allow it to incubate. If there's phenylalanine in the blood, B subtilis will grow near that filter paper with the patient's blood on it, and it won't grow anywhere else. So that's one of the ways that we test for phenylketonuria. For other types of amino acid analysis, blood samples should be drawn after at least 6 to 8 hour fasting. So the patient should fast for at least 6 to 8 hours. Sample is collected in a heparin tube, and the plasma is removed promptly from the cells. So as soon as we collect that, we are going to mix it with the heparin, and then we're going to centrifuge it, and we want to remove the plasma. While we're removing the plasma, we want to make sure that there's no hemolysis, and make sure not to remove the platelet and white cell layer of that tube. Then we do deproteinization within 30 minutes of sample collection, and analysis is performed immediately, or the sample can be frozen at -20 to 40 and performed later. Urinary analysis is performed on random specimen for screening, and then the method of choice for screening is thin layer chromatography. So now that we've covered amino acids are going to come together to produce proteins. So we're going to jump right into talking about proteins. Proteins are synthesized and catabolized in the liver, so they're created and broken down predominantly in the liver. The building blocks of proteins are amino acids, and those are linked by peptide bonds to form polypeptide chains. And there are 4 structural levels of protein structure. So we have primary, secondary, tertiary, and quaternary. Primary structure is talking about the sequence of amino acids. So when we first produce the protein, we are going to get directions from the DNA, and the DNA consists of genes, and those genes are going to consist of codons, and codons are going to code for a particular sequence of amino acids. So that sequence of amino acids, those amino acids that are linked together by those peptide bonds, that's going to be our primary structure. Amino acids have all of these side groups, and those side groups are going to be interacting with each other as this polypeptide chain is produced. When those side groups form hydrogen bonds with one another, they're going to produce secondary structure. So secondary structure is a 2-dimensional folding of the protein, and the 2 most common secondary structures are going to be the beta pleated sheet and the alpha helix. When those secondary structures kind of start to fold and bend together and hydrophobic interactions come into play and we start to take on more folding, it becomes tertiary structure. So it's the interaction of alpha helices and beta pleated sheets within a single polypeptide chain, and that's going to create a 3-dimensional or conformation to the protein. So 3-dimensional shape or conformation of this tertiary structure. Quaternary structure is when we take multiple tertiary structures and combine them together to produce a complete protein. So multiple tertiary structures coming together to create a larger oligomeric structure is going to be our quaternary structure, and this is our functional protein. So as I just mentioned briefly, proteins are synthesized through the process of transcription and translation. During transcription, a messenger RNA copy of the DNA is produced. That messenger RNA is modified, introns are removed, and that messenger RNA will then travel to a ribosome in order to start translation. During translation, the ribosome is going to read the messenger RNA and incorporate certain amino acids using transfer RNAs, depending on the sequence of codons that are found in that messenger RNA. If you're unfamiliar with the process of protein synthesis, you should review your notes and review previous content to kind of get caught up to transcription and translation. Protein catabolism, or breaking apart proteins, is another component of human health, and this is something that we're going to talk about in a future lecture when we get into non-protein nitrogen and urea wastes and kidney function and all that type of good stuff. So when proteins are broken down, they're broken down by separating amino acids from one another. So when we break apart an amino acid, we do that with hydrolysis. If we look at that word hydrolysis, we have hydro and lysis. Hydro is water, lysis means to break apart. So we're literally breaking apart the molecule using water. And you can see here the bond that existed, and when we incorporate water into that bond, it breaks it into 2 separate parts. From there, the amino acid undergoes deamination. Deamination is the removal of the amino group from the amino acid. So when we rip off that amino group, it creates ammonia, and then there's the organic acid. The organic acid can be used in glycolysis or the citric acid cycle, also known as the Krebs cycle. So right away, this one can be incorporated into making energy or producing different types of amino acids. But this ammonia is a problem. Ammonia is toxic. It makes us sleepy and it can cause brain disturbances, and we need to get rid of ammonia quickly. So ammonia is going to go to the liver, and we're going to produce ammonium and then eventually urea. And urea, while still toxic, it's less toxic. So we can handle urea being around in the body a little bit better than we can handle ammonia. So urea is produced in the liver, and then it's going to be eliminated in the urine.